Okay, welcome back, guys. We're forging our way into these prokaryotic cells, and specifically we're looking at more so bacteria than archaea. And we've now already kind of finished the prokaryotic envelope. So in other words, we've taken this area out here with all of its extensions, its fimbriae, flagella, whatever else is associated with that cell wall. Sometimes you're going to have a outer membrane with a gram-negative organism, and we're past that. We're now forging down into the cell and looking at what we'd find inside any cell, any prokaryotic cell. Now, to some extent, we have to remember that these cells are living cells like any other, like any eukaryotic cell in ourselves. So they need all the same things. They need, for the most part, most of them need oxygen. Not all, but many need oxygen. They're going to have a gas exchange going in and out of the cell. They need food, right? They need energy to do work, and they need to replicate themselves. So they're going to have a lot of the same basic functions. It's a living cell. And when we move into the cell, we're going to see some of the same structures. Cytoplasm is basically everything inside of a cell. It's all substances within the plasma membrane. And here we're talking about the inner membrane here, right? Not an outer membrane that would be associated with a gram-negative. So everything inside that plasma membrane is cytoplasm. And the big difference with a prokaryote versus a eukaryote is that eukaryote actually means true nucleus, okay? There is no true nucleus. This is prokaryote. And so within a cell, a prokaryote cell, you're not going to see a separate set of compartments, including a nucleus. We're not going to see that, okay? What we're going to see is kind of a little bit more diffuse existence of the bacterial genome, or the chromosome, which is basically the DNA, right, the genetic material. And it's going to take up a good part of the inside of the cell. So here we see something called nucleoid, and you see here in the dashed green line down below that nucleoid exists, kind of taking up a good part of the center of the cytoplasm. Now, this look will change when the genetic material is being replicated. And we can see, if you look closely, that there are actually a few places here inside the cell. Let's use red just so it shows up. Here over on the right, we see this circular piece of DNA that is similar to the nucleoid in that it is circular, okay? But it's much smaller, and we call that a plasmid. We're going to be running into these plasmids here in a little bit later, okay? So plasmids are pretty unique to prokaryotes. We don't see plasmids so much or talk about them in eukaryotes. But we have this circular bacterial genome. All of the genetic material in a eukaryote is linear, not circular. And there's certainly more than one of them in most eukaryotes. So if we look at that, that's another difference, is there's one circular nucleoid bacterial genome. Now, this, like any other genetic material, is made out of nucleic acids. So you have your nucleotides, right, which are the building blocks. We've done that in the chemistry assignment just to kind of refresh some of those ideas. These are the monomers, I guess you would say, of the DNA. Okay, and this whole chromosome would be a polymer of all of those. Now, we're also going to see inside the cell these little dots, which happen to be ribosomes. We're going to see a place where we'd have some kind of protein synthesis going on. Okay, so proteins play the role of enzymes sometimes. They're structural. They give structure to things sometimes. They have multiple roles, and this is a living thing that needs those kinds of things just as much as any other. Okay. Now, one big difference again that we see here between the eukaryote and the prokaryote is that there's a different size of ribosome, right? So in eukaryotes, we see 80S. Think of S as size. It really means Svedberg unit, which you don't have to worry about too much, but when they were actually identifying these structures within cells, they had a different rate of diffusion through a porous material, and they gave them those weird units. But basically 70 means it's a smaller size ribosome. A smaller protein factory is found in a prokaryote than is found in a eukaryote. Now, you'll notice as we move on here that I'm stressing differentiation. Early in lab, we were differentiating based on macroscopic, what they look like on a agar plate. Then we went microscopic, and we wanted to differentiate the prokaryotic cells, the bacteria, from one another. And we even saw in lab how much smaller they were in comparison to eukaryotic cells. So again, differentiation, we want to tell them apart. Now we're looking at subcellular structures. We started with the envelope, and we saw some very unique structures, peptidoglycan layers, things like that, not found in eukaryotes and not found in plant cells either. And now we're seeing differences inside the cell as well, always trying to tell them apart. Okay, this will become important later, because if we want to kill certain kinds of cells, like in the clinical setting, in a aseptic kind of lab setting, or in a medical setting, right, where we want to get rid of a bunch of cells that could be pathogens, we want to be able to tell them apart so that we can kill them very accurately, right? We don't want to introduce chemicals or things like that that would adversely affect our own cells. We want magic bullets, okay? Now, as we move into these cells a little further, we start to see that there are other subcellular compartments that aren't necessarily membranous always. That's more to do with eukaryotes, right? Eukaryotes have these membranous compartments that we'll talk about again later. But prokaryotes don't have a lot of those kinds of compartments, with just a few exceptions. We talk about inclusions when we look at prokaryotes. So when we look at the inside of prokaryotes, we see inclusions that would have to do with energy. So a storage that would give energy to a cell later on, polysaccharide granules. Now this is something that we do sometimes see in eukaryotic cells, because polysaccharides could exist in the form of glycogen. And in fact, the human liver is packed full of glycogen inclusions. Okay, so some of these are found in eukaryotes, but it makes sense that a sugar, a polysaccharide, would be a good energy reserve. We have lipid inclusions inside of prokaryotes. Again, that's all about energy storage, right? We had to think of functions of these macromolecules, and energy storage would be top of the list when I think of lipids. Sulfur granules. You may not think of energy when you think sulfur at first, but sulfur is involved with energy production in many prokaryotes, and we'll talk about metabolism later, so that makes more sense. We're going to find that there are some gas vacuoles, like you see over here in these little pockets here. There is gas, and believe it or not, organisms like this can produce more or less gas vacuoles dependent on when they want them. Let's say that this cell is a photosynthetic cell, so it tends to have chlorophyll, maybe like plants, and it likes to be close to the light, right? So it wants a phototaxis, and it wants to go towards the light. If this is a cell in an aqueous environment, it could make more of these gas vacuoles to help it to be more buoyant and come to the top. More light at the top during the day, and during night, it'll get rid of some of those gas vacuoles. These cells don't have a brain. They're not aware, like we are, of what's going on, but they're very smart, and they've evolved over millions of years, so we have to give them credit. They're doing some pretty sophisticated things here, and we haven't even scratched the surface of what many of these bacteria do. Okay, a last one that I'll mention is magnetosomes, which are really kind of cool structures. And I have here listed magnetosomes as a protective function, which I have read in some resources, but I'm also reading that this can also provide for motility. So again, movement around based on magnetic fields. So think of this. This looks like a kind of a curved bacterium, right? A lot of times, it's not quite a bacillus organism, but we see these magnetosomes kind of in an alignment here. Think of this like a compass needle, right? They found that these magnetosomes arrange themselves in such a way that it will help these cells to move in relation to the magnetic field. We find them in aqueous environments. It's really fascinating, and again, we don't know half of what these things do probably, okay? Just a couple other pictures here. This is straight from chapter 3, I think it's 3.3, where they talk about some of these things. Here they've got some lipid inclusions. Over here, I think these are gas vacuoles. These are definitely gas vacuoles here. Okay, and then here we see some of those magnetosomes, right? All kind of lined up here in this particular cell, helping it to align with magnetic fields. Okay, the last thing I want to talk about in this clip with you guys is something called an endospore. All right, so here we have a basic set of steps of sporulation when it comes to, now we're talking about bacteria. Keep in mind, this is all prokaryotes we're talking about now, so you might have heard of spores associated with a fungus, fungal spores, and absolutely they exist, and there's something we could talk about, but right now we're talking about spores that would form from prokaryotes, from bacteria, okay? So what I have in an image here on the right side is an organism called Bacillus. I think this is Bacillus subtilis, which is just the species name, okay? And this is growing on a TSA plate. So I can see there's individual colonies here. You can see these are not perfectly circular colonies. I can see they're white for sure, but they have the ruffled edges here and pretty interesting looking cells, colonies at least. Now if I were to let these cells sit on a petri dish like this with food, right, because the agar has nutrients, and let it sit in the incubator for 4 days, not just 24 hours like we typically would do, find our little colonies, right, but 4 days, what these cells would do is they would very quickly fill the surface, and they would be in great competition with one another, okay? They would actually have found a state where they need to conserve on energy because they're competing for the very same food and same space. So what this culture would ultimately come to is a resting state, okay? It's not actively growing as a population. It's kind of chilled out a little bit. It's just these cells are kind of waiting for more food to come, all right? And in a resting state where there's a limitation of nutrients like this, what cells that have the ability to sporulate will do, okay, is they will replicate their DNA. So number one, they're going to replicate that DNA, okay? This is similar to what cells will do when they are going through rapid division and growing in population. But this replication of DNA is not for that purpose. It's not going to go through this replication process to make 2 cells. What will happen is a membrane will form around this replicated DNA. Okay, so that's step 2. Eventually, an additional membrane will form around that DNA, okay? And this will form what is known as a septum, a double membrane around that replicated DNA. Then peptidoglycan will form between those membranes within that septum. Okay, this is a protective cortex that will form. And then outside of that, we have a protein coat, a spore coat that will form. And the spore will be released. And this spore is, you think of it almost like a seed. It's not actively growing. In fact, it's resistant to lots of different conditions. It doesn't need nutrients. It can resist lots of different environmental pressures in this state that the living growing cell would not have survived. All right? Here we see a microscopic view of vegetative cells. Okay, so these would be the cells in red. So this would be a vegetative cell over here before it gets started. This is a living growing cell, right? And then in green here we see these little compartments, which are basically these endospores here that are getting ready to be released. Right? As endospores, as spores, they can sit there for a long time. Okay? Now, when you boil water, let's say, or boil chicken broth or whatever it is, you're going to kill a good number of bacteria because heat kills cells. Okay? It's going to denature proteins, going to kill cells. But unless you achieve a certain temperature, these spores are going to stay around. Right? Just boiling water is not going to kill these. This is why you need high temperatures and pressures, like in the lab where we have something called an autoclave. We need to autoclave things to get rid of these spores. Okay? These spores, like a seed, could germinate and turn into vegetative cells. All right, so a little bit more on sporulation, excuse me. This process is going to be found taking place in resting cells. So these cells have reached a point where they are fighting for nutrients, right? There's a limitation when it comes to nutrient levels. But what are these spores capable of being resistant to? They're resistant to dry situations, desiccation. They can't really get dried out like a vegetative cell could. Temperature extremes, really hot, really cold. It can take quite an extreme set of temperatures, okay? Unless we go super high in an autoclave. These would still survive. Caustic chemicals, right? Chemicals that would kill the vegetative cell will not kill a spore. I've also gone online, and I'm looking at some of my notes here. Radiation will do something to vegetative cells, but a lot of spores will survive. Freezing, we talked about. Temperature extremes, they'll survive that. In fact, I have in my notes here that some spores have remained viable for as many as 750,000 years. They found spores from certain like freezing muds that are in lakes in Minnesota where the spores germinated that many years later, given the chance. They found spores in 25 to 40 million year old amber samples that they found. Okay, these are viable spores. They're amazing, really, that they can stay viable like that. Now, I was saying earlier that there are some tricks that gram-negative organisms had, these fimbriae and pili and things that they can do. Well, this is primarily a gram-positive trick. These spores are found in gram-positive organisms. Bacillus is a genus with many species, okay? Something I should have probably mentioned earlier. Any genus name is italicized, okay? And then the species, and I'm not being specific here, I'm just saying species, several different species within this genus group of Bacillus. And down here below you see a few listed, right? Subtilis, Sirius, it's kind of a weird name, right? B. Sirius. But these we use in the lab all the time. But if you look at some of these other names, they might ring a bell. So B. anthracis causes anthrax, right? Now, if they're in spore form, they're going to resist a lot of different conditions, right? This organism in spore form is something that they were worried about with bioweapons, right? With bioterror. The fact that you could take these spores and have them, God forbid, be released in the vents of the subway or something. These were what were found in some letters that were sent around to a few politicians, I think. I don't know if you were even alive then, some of you, but probably were. And these were pretty big scare, because this can kill you, right? Anthrax is pretty dangerous if it gets into the lungs. Another genus, and you see it here, is Clostridium. So let me give you a few names within the Clostridium group. Clostridium tetani, tetanus, right? Clostridium perfringens. Now that one doesn't really tell you in the name, right? It's Clostridium perfringens, but Clostridium perfringens causes gas gangrene. People lose fingers and toes to gas gangrene. Another one I don't have here, but one that I'm thinking of is Clostridium botulinum. It causes botulism. Okay, it paralyzes muscles, can kill you if you get food with Clostridium botulinum in there. All right, another one, let's see, C. diff, right? Clostridium difficile. This causes major issues in people's guts, and these spores are in our guts in healthy people too. But what antibiotics do is they kill a lot of the favorable bacteria that help us, and then these spores become active, right? Now there's plenty of food to be had. They're not outcompeted. This C. diff can get going, right? So after long courses of antibiotics, these are things we worry about. All right, so we talked about sporulation, but keep in mind you can also go the other direction, right? Germination. So this would be a spore returning to its vegetative state. We can go both ways. If there's favorable conditions and more nutrients, a spore will become a vegetative cell. And so we'd be going in this direction, right? Going from a spore that's blue here to a vegetative cell, instead of going the other direction to form the spore. All right, so I think this is a good place to leave off, and next time we'll kind of launch into some other major differences between these prokaryotes and the eukaryotic cells that make us up. Okay?